So I'd like to introduce our seminar speaker today. This is Melinda Connors, our local albatross whisperer. And a little background on Melinda. She spent her bachelor's degree time at UC San Diego, right? And then she was a quote-unquote field junkie for a long time. She spent a lot of time on the Northwest Hawaiian Islands, at the Farallones in Alaska, out in the field. And she's currently a doctoral student at UC Santa Cruz studying albatrosses, and her talk today is going to be the characterization and interpretation of the diet of North Pacific albatrosses using quantitative fatty acid analysis. So thanks, Melinda.
Cool. Thank you very much for the introduction, and thank you all for coming to the talk. It's great to see all of you here. As Ryan said, I'm a PhD student up at UC Santa Cruz, and the stuff I'm presenting to you today is most of my second chapter, but my dissertation as a whole is on the foraging strategies of North Pacific albatrosses. In particular, using movement data, diet data, and behavioral data to investigate these foraging strategies. So today I'm going to talk mostly about diet, so I used a sort of recently developed biochemical analysis on lipids to characterize the diet of these birds. So I'm going to talk about that. I'm going to talk about what diet means in the context of foraging strategies, and then I'm going to talk a little bit about what variability in diet means. Okay, so we'll start here with wolves, as you do. And as you can see on the slide, there's this very hungry pack of wolves that are taking down a particularly worried-looking deer in this very nice forest setting. What you don't see on the slide, however, but what comes to mind for an ecologist looking at the slide is all the complex relationships between these wolves and the deer and the forest that they live in. And so terrestrial ecology there's been a lot of studies in terrestrial ecology about predator foraging strategies, predator-prey relationships, and trophic web dynamics. And so some really nice studies have come out showing these intricate relationships. For example, wolves have a positive impact on aspen groves by reducing the grazing intensity by the herbivores, because they're reducing herbivores in the environment. But what about the marine ecosystem? So obviously there are lots of large predators in the seas, but the effects that they have in modulating their environment are largely a mystery. And this mystery only gets deeper with the modern collapse of fisheries, shark and whale populations, as well as many other marine taxa. So sadly, marine ecosystems are degrading before we can even begin to understand them. For most ocean predators, we don't even know what they eat, much less the impact that they're having on other animals in that ecosystem. But don't despair, some good work has been done. So the seminal work of Jim Estes brought terrestrial ecology concepts into the marine world with his great work on sea otters foraging along the coast of North America. And so to scientists and non-scientists alike, we all know his work for showing that a decrease in otter populations due to predation by orcas led to an abundance of sea urchin, which then led to a catastrophic decline of kelp in these coastal ecosystems. And so these species were coined keystone predators for their very influential role that they had in modulating their environment. Okay, and so Jim Estes and Tim Tinker have continued this work with sea otters, and much of their recent work has focused more on individual variability and foraging strategies. And so some beautiful work has come out of this, showing how otters actually select certain prey over other prey, so they have prey preferences, and these preferences not only stay with them their whole life, but they're also passed down from mom to pup, so along the matriline. Additionally, this paper in Ecology Letters by Tim Tinker showed that competition for resources also has effect on the individual variability of diet in a population. So in sea otter populations that have lower densities, most of the individuals are all foraging on sort of the preferred dominant prey type because there's not that much competition. But as you increase the density of these populations, you get clusters of individuals specializing on more marginal alternative prey types. So the take-home messages here, one is that individual variability is not just random noise in the environment, but in fact, in this example, it's well regulated by both intrinsic factors, such as the matroline, as well as extrinsic factors, such as the density of the sea otter populations. And furthermore, variability in a population is important on evolutionary timescales because it serves as sort of an edge in the population that natural selection can grip and pull that population in particular directions. So the amount and characteristics of variability in a population contribute to the capacity for change and evolution within that population. So there's all this great work that's been done on sea otters, but sea otters are not your typical marine predator because they're highly accessible. You can watch them from shore. You can see what they eat because they bring their food to the surface, and so all you really need to observe them is just a good pair of binoculars. That's not the case for most marine predators that impose a lot of limitations and their own set of challenges in trying to observe. But again, progress has been made, and this paper actually just came out this week, and it's a really cool paper showing how one marine predator, pink salmon in this case, can have a huge impact on other marine predators in the environment. And so what they showed is, well, this is basically a bastardization of the results of the paper. And what it shows is, so pink salmon have a 2-year life cycle. And so every 2 years, you get this huge influx of hungry pink salmon into the subarctic, Pacific, and Bering Sea ecosystem. And when you see these large peaks in abundance of salmon, correspondingly you see these drops in reproductive success in the seabirds. And so what the researchers suggest is happening is that, well, they know that pink salmon forage on similar prey types as the seabirds, so they're concluding that the salmon are basically outcompeting the seabirds. An important thing to recognize here is that to make that mechanistic link between the fish and the birds, you have to know what they're all eating. For birds like salmon and kittiwakes, characterizing their diet is more akin to characterizing the diet of a sea otter, just because they're pretty accessible birds and they forage on a short time scale. So in the case of a puffin, they're bill loaders. So as a researcher, you can basically just watch them fly in with the food that they foraged on in that particular day and identify it. With kittiwakes, they're foraging, they go out for the day, they come back, and the food in their stomach has not been all that digested, so it's easy to identify as well. But what about the species that you never really see? And if you do, it's just sort of a lucky brief moment in time. Most predators in our ocean ecosystems are far-ranging, unobservable, and are foraging in highly inaccessible regions of the oceans. So how are you supposed to characterize the diet of something that you can't even see, much less watch eat? And in the case of the puffins and kittiwakes from the last slide who are foraging on shorter timescales, things like albatross and northern elephant seals are foraging for weeks, if not months on end, integrating diet into their bodies as they go along the way. So this presents a unique challenge in trying to understand the diet of these very important large marine top predators. That's not to say we don't know anything. So most of what we know comes from content analyses, and how this works is basically by whatever means the researchers get their hands on stomach content of these animals, whether it's by opportunistically collecting a carcass that washed up, or using stomach lavage to induce vomiting. But since they're out foraging for such long periods of time, usually all that's left in their stomachs by the time you sample them are hard, indigestible parts like squid beaks or fish otoliths, which are the robust ear bones of fish. And this is awesome because you can actually identify the species. You can actually identify species based on their squid beaks and otoliths. So using these hard bits, you can actually reconstruct the diet of these predators. But there is a limitation to this, because what about all the soft-bodied prey that don't leave behind hard bits? And there are also species of fish like myctophids, which are hugely abundant across the North Pacific, that have smaller otoliths that readily dissolve. So this is an important technique, but it's also leaving a lot of holes in the diet data. So recently, a lot of more modern biochemical techniques have been developed to sort of fill in these holes, like stable isotope analysis, fatty acid analysis, and even a DNA barcoding of prey. So we'll go back to the albatross and northern elephant seal, and hopefully I'm convincing you all that they're good candidate species for some of these biochemical analyses, given their elusive and inaccessible natures. Additionally, they're good candidates for this kind of work because during certain parts of the year, they come back to land for important parts of their life history, such as breeding and molting. And these events are highly synchronous, very predictable, so it's easy for the researcher to know when to go out there to access these animals. Furthermore, as all living things, as most living things, well, all living things, have lipids in their tissues that the researcher can sample and then compare or use in a fatty acid analysis. So just briefly, what fatty acid analysis does is basically it works on the premise that all marine organisms, most marine organisms have characteristic lipid signatures, kind of like a lipid fingerprint. And then you can use a library of prey lipid fingerprints and compare it to the fats inside your predator, and then use a model to give you sort of a best estimated diet. Albatross are pretty cool because they are in a family of seabird species called the tube noses, and they actually have a unique adaptation to a life in flight by converting all of their prey that they consume into this highly energy-rich oil, stomach oil, which is thought to be an adaptation to sort of lighten their load for migrating long distances. The beauty of this is that this oil is derived directly from prey. So an elephant seal, where when you sample their lipids, it's incorporated into their tissues. It's undergone metabolic transformation, so you have to know how the lipids change from the prey into the tissues. But it's definitely doable, and my lab mate, Chandra Getch, is actually simultaneously, as I've been working on the albatross project, she's been working on reconstructing the diet of northern elephant seals using this technique. So this is sort of where my story starts with my project. My project stemmed off of a much larger project called Tagging of Pacific Predators, which was an effort to put a lot of electronic devices on a lot of different marine predators and see how they're using this vast ocean environment. And so my advisor managed the albatross component of this project at Tern Island in the French frigate shoals of the northwestern Hawaiian Islands. And so people have been out there tagging lace-sand and black-footed albatross since 2003 or 2004. And so by this point, we have huge amounts of tracking data and a very good idea of how these birds are using their environment and the habitat preferences that they show. So on this slide, let's see. This slide shows a subset of that tracking data. So this is about 3 years, I think 3 years of some of the GPS data that I collected at Tern. And so the breeding colony is down here. And as you can see, these birds are using an enormous amount of the North Pacific. And an additional thing to note is that the Laysands, who are colored by yellow, are found more on the northern border of this highly productive North Pacific transition zone, which is a good place to eat if you're a seabird or a marine mammal. Whereas the black-footeds in red they're preferring more of the southern, more warmer waters of the southern edge of the transition. And also exploring some of these more southeasterly waters. What you can't see from this, which is incubation and brooding tracts, is that, so these are all the brooding tracts, and it just looks like a big mess of red and yellow points. So a really interesting thing is that during incubation, when these birds can go out and forage for 3 to 4 weeks at a time, there's distinct habitat segregation. But during the chick brood period, all of a sudden you have this extreme range reduction, and now not only are they limited to foraging in suboptimal tropical waters, but they're also experiencing an extreme increase in competition, not only within their own species, but also between the species. So how they're doing this and how they're mitigating this competition is definitely one of my focuses in my thesis. Okay, so we know that when they're not in the chick brood, at least, there are distinct habitat preferences. But what about diet? So we do know some about their diet. We don't know nothing. And so what we do know mostly comes from, this is sort of the only comprehensive diet study from breeding albatross from the Northwesterns. And so this, not the only one, but, well, anyway, I won't get into that. So this paper, which came out in 1983, was based off of chick stomach contents during the chick rear period. So when the chicks are getting older and the parents can leave them and travel farther at sea. The researchers basically puked the chicks and saw what was inside the chicks. And what came out of that study was that Laysan albatross were utilizing mostly squid, and black-footed albatross were utilizing mostly fish eggs. So sort of the take-home message from that paper was, well, Laysan albatross also have high levels of rhodopsin, which is a light-sensing pigment in their eyes. They're foraging on these emestrophid squids, which are known to vertically migrate and be at the surface at night, so they must be nocturnal squid specialists. And blackfoots, on the other hand, are eating all these flying fish eggs, so they must be flying fish egg specialists. But then this came out, and this was a poster presented by William Walker from the National Marine Mammal Laboratory about, I think, 3 or 4 years ago at Pacific Seabird Conference. And so what you don't know about this study is that over 90% of the squid remains in the stomachs were squid beaks, and they were left unidentified. So all the squid, that's a huge component of their diet, was identified just on few undigested individuals of squid. And what Bill Walker did is he took all those squid beaks, identified them, and found out that the vast majority of them are actually from mature, large, deep-living mesopelagic gelatinous squid. So these squid never see the light of day. I mean, they live deep, they don't vertically migrate. So how on earth is an albatross getting a squid that lives at 1,000 meters of depth? So the same squid beaks are found in elephant seal stomachs, is really interesting because albatrosses obviously can't dive as deep as an elephant seal. So his hypothesis is that since all of these squid beaks came from mature adult squid, he thinks that these squid spawn, they die, and then a lot of them are known to be positively buoyant after death, so they float to the surface and then are scavenged by these seabirds, which presents a very different story to their foraging strategy than them feeding on live squid at night at the surface. But we still don't know quite a bit. So we know a lot more now about the squid that they eat, but what about the soft-bodied prey like jellyfish, crustaceans, and even myctophids, which are those fish that are highly prevalent in the North Pacific transition zone, but have small otoliths that easily degrade. What about them? How does diet change across time? So we don't know how it changes across the breeding season, and they're foraging in very different areas across the breeding season, so that likely affects their diet. And we don't have individual-based diet measures. So thinking back to Jim Estey's work on sea otters, we don't have individual-based diet, so we can't answer any of those questions. So the objectives of my work were, one, to characterize the diet based on one of these biochemical techniques which attempt to go around these limitations in traditional stomach content analyses. To put diet in the context of competition, so comparing diet during incubation versus the brooding season where they're experiencing more resource competition. And also to get some individual-based diet measures such as individual variability, to look at group differences, so differences in age or sex, and also to look at dietary specialization. So are these birds just eating a lot of stuff, or are they choosing a strategy? Do they have a preferred prey like the sea otters do? Okay, and so on to QFASA. So let's see. So to do quantitative fatty acid analysis, it requires three main things. One, lipids from the predator. Two, if you're going to take lipids from a tissue like an elephant seal, you need a calibration coefficient to deal with the metabolic transformation. And three, you need a prey library of fatty acid signatures. So for predator lipids, again, albatross are really, really great species to do this method because they have this stomach oil that you can collect that's derived directly from prey. And to do that is actually relatively a quick and simple procedure. You just slide a catheter tube into the stomach of the bird and pull up a small aliquot of stomach oil in a syringe, and then you have your diet sample. And so I collected, from a subset of birds that I tracked on Tern Island, I would collect a stomach sample at the end of that trip, and that would represent diet integrated over most of the trip. Okay, then once we have lipids, you need to extract and purify the lipids, and what you're seeing here is an actual albatross stomach oil. What's interesting is take note of the variability in color, and that's the first good indication that there's a lot of variability in diet because this pigment is probably coming from the pigment of their prey. Two, then I separated the lipid classes. So typically in fatty acid analyses, they mostly focus on triacylglycerol, and that's because in tissue, triacylglycerol is a structural component in things like blubber and tissue. So the reason I wanted to separate out lipid classes is because since I was getting lipids directly from prey, I knew that marine prey tend to have a lot of wax esters. We were seeing a lot of it in the diet, so we thought it would sort of double the strength of the model if we included both lipid classes. So to do that, I had to separate out the different kinds of lipids in this technique called thin layer chromatography. And then once I had my different lipid classes, I had to break them up into their fatty acid and fatty alcohol components with a lot of chemicals. Once you do that, then you can plug those into do some gas chromatography, and the output is basically your lipid fingerprint of that particular sample. Again, didn't need a calibration coefficient, so the last thing I needed was a prey library. This was a daunting task, seeing that, as you saw, these birds are covering pretty much the entire North Pacific Basin and could be eating anything. And the prey library is a very important part of the model because if you're missing a key component, it could really just estimate a really wacky diet. So I had to come about this with some strategy, and what I decided to do was to try and not think of it as individual species of prey, but think of them more as functional groups, and then go out and try to collect representatives from those different functional groups. So I'll go through the different functional groups that I targeted right now. So the first two were vertically migrating muscular squid and small schooling epipelagic squid. And so these are the squid that you and I are most familiar with, market squid we eat, squid we see at the surface. And a lot of these smaller guys are also bioluminescent and surface at night and have been found in stomach contents of many seabirds, so I felt it was important to represent them. Also, of course, these gelatinous meso to bathypelagic squid, so these super deep living guys that don't come to the surface but are showing up in all the squid beak studies. Really cool squid. And then also vertically migrating mesopelagic fish, like myctophids that I've been mentioning, and crustaceans. So these are things living in this thing called the deep scattering layer. Basically, it's this habitat where a lot of these smaller, deeper living prey items live, and during the day, they inhabit fairly deep waters, but during the night, they actually come quite close to the surface, and many surface predators can access them at night. I wanted surface Newston like jellyfish and Velella and salps. I thought this was pretty important since you're not going to see this prey represented in stomach contents because these are just going to disappear right away with digestion. Obviously, I needed fish eggs. I also wanted to get long line associated foods such as bait and target prey because we all know that seabirds love to follow fishing boats, and it's well known that albatross supplement their diet with fisheries-associated food. And then I needed some surface schooling epipelagic fish that these birds might be getting with the help of subsurface predators. And finally, I wanted to include marine mammal blubber of some kind because there are lots of live things in the ocean, and there are also lots of dead things in the ocean, and these guys float at least at some point after death. Albatrosses have been known to scavenge carcasses, and they're highly attracted to the smell of fat, so I thought I needed to include some blubber in there. Okay, so how did I get all those things? I was actually really lucky. My lab mate Chandra and I were able to go on a Japanese research vessel that transited into the North Pacific Transition Zone and conducted midwater trawls and surface tows as well as squid jigging. So I got a lot of prey from the North Pacific Transition Zone from this trip. But I also needed prey representing more local waters for the chick brood period when they're restrained to Hawaiian waters. And so I was fortunate to get some samples that were frozen in a NOAA lab to represent that period of time. And then I also contacted a bait supplier in Honolulu. So they supply the long lines in the area, and they were able to give me some bait specimens that they use to supply the long line fleets. And this bait is sourced from Asia, so Pacific Sori and chub mackerel from Asia, and so they should have a very different lipid signature than any of these sort of larger teleos fish that the albatross would encounter more in their typical foraging range. So here's my prey library. Don't worry about reading a thing. All I wanted to show is that I got 33 species, and all these different colors represent the different functional groups. And so I was able to get representatives from each of the targeted functional groups that I wanted. So next, this is the funnest part of everything. But basically, you just do the same thing with the prey samples that you do with the predator samples, only before you do that, you have to basically put it through a giant meat grinder, which was fun. And then you have your 3 requirements so you can run the model, which is just the easy, you just press the button. Oh, it's not that simple, but yeah, you run the model and you hope that it makes sense. And so now I'm going to go through the results. I'm going to start with diet characterization. So I know there's a lot on here, but I feel like it's important to just say what the model said they're eating. So this is sort of the raw output, and I'm going to guide you through it in the next slide, so don't worry about getting it all right now. But basically, all my samples are on this axis, and then percentage in diet is on this axis. All these different colors represent different birds, and this is the individual diets that are showing up per prey species. So for black-footed albatross diet, I'm going to sort of go through my functional group. So small epipelagic squid, so those guys that are likely being seized at the surface, possibly at night in a nocturnal strategy, are showing up in the diet. Not hugely significant, but they're definitely there and important to note. Mesopelagic fish is coming out really strong, which is surprising if you think about Harrison's hypothesis that black-footed albatross are just diurnal scavengers, but they're showing large amounts of myctophids in the diet. Some individuals showing high individual specialization on these prey. Then we have a lot of mesopelagic gelatinous squid as well, not a surprise. Again, some individuals really specializing on them. And then muscular squid. This is interesting because I collected both adults and juveniles of some species, and they were not by any means going after the juveniles. So only the adults really showed up in the model, suggesting that for them too, they're probably acquiring them on a post-spawning basis. So these individuals also spawn and die, and then they're probably scavenging these carcasses rather than feeding on them live. Longline associated prey, bait species weren't showing up too much. Swordfish was showing a fairly strong presence. Crustaceans were the big surprise for me. I mean, I knew they were in the diet, but it's a lot stronger than I expected. And so at first I was a little concerned that maybe something was off, and maybe something is off. But after doing more research and thinking more about it, I feel a lot more confident that this is actually the case, that they are eating lots of crustaceans. Note that they're not eating krill, copepods, mysids. They're really hammering these large pelagic decapod shrimps, or something that has a very similar lipid signature to that, like some sort of pelagic crustacean. And luckily, I came across this paper from 1940, like 3 days after I ran this model, and was like, what's up with these shrimp? And I'm just going to read this to you because I thought this was awesome. Okay, so Loy Miller, 1940, from the Condor. On one of the cruises, large numbers of a brick-red decapod crustacean were observed coming to the surface, presumably from deeper waters. These animals swam slowly about at or near the surface, either in spawning activity or perhaps as victims of an infection that reduces their specific gravity to a point less than that of seawater. In the course of this decapod swarming, albatrosses seemed to avoid a great volume of excrement of brick-red color that could surely have had no other source than the crustacean for its carotenoid pigment. And thinking back to the colors of those stomach oil samples, a lot of them are really bright red. So to me, it seems like that's not too out there of a result, that they really could be hitting up crustaceans more than we would expect. And then sort of my other category, so fish eggs, very, very high in some individuals, not as strong of a presence as I expected, but definitely there, and a little bit of marine mammal blubber. No jellyfish, no small epipelagic schooling fish. And onto laysand diet. Okay, so again, small epipelagic squid showing up a little bit more strong in them than the blackfoots, which might support Harrison's study more, that maybe they're going after these guys. They're foraging more at night than blackfoots potentially. Small epipelagic fish are showing up, but nothing significant. Mesopelagic fish also very strong in the laysands. Mesopelagic gelatinous squid very, very strong in laysands. What's interesting is that the species of mesopelagic gelatinous squid are different between laysand and blackfooted. This might be due to the fact that these guys have very distinct habitat preferences, and it's dictated mostly by sea surface temperatures. Squid are known to have very controlled spawning behavior based on sea surface temperatures, so potentially, because the birds are segregating by sea surface temperatures, they're maybe just not encountering squid of the other species. Another thing to note is that I've been talking a lot about mesopelagic stuff, and so it's just important to think that there's this huge amount of biomass deep in the ocean that is turning out to be very important in the diet of these surface birds, and just keep that in mind. Muscular squid, again, only the adults are really showing up. This is interesting. This was a paper showing that the spawning ground for neon flying squid, some of these large vertically migrating squid, actually occurs right around the Hawaiian Islands during the time that albatross breeds. So potentially the albatross are exploiting this ephemeral abundance of post-spawned squid. And then longline associated, also showing up more strong in laysand than blackfooted, and interestingly enough, it's unlike the blackfoot that weren't showing any presence of longline bait, the laysands are, whereas the target species aren't showing up as strong. So it seems like the two species might be exploiting longlines in different ways. Again, crustaceans showing up, and one bird went for some fish eggs, and then a small smattering of marine mammal blubber showing up. Okay, so now I want to kind of move away from thinking about individual species and think more about these functional groups. So this is basically how these functional groups separate out in statistical space based on their fatty acids. And so the point of this slide is just to show that, that they do group by functional groups. So epipelagic fish and epipelagic squid group together. The mesopelagic guys cluster together. Interestingly, and it's good to see the marine so this is elephant seal blubber, and it's clustering with the mesopelagic guys, which makes a lot of sense because they eat a lot of myctophids and mesopelagic squid. And then we've got the crustaceans over here, so krill and copepods, and then mysids and shrimp. Now to think about diet in terms of functional groups, going back to, and I also want to transition to thinking about how diet changes across the breeding phase. If we start with black-footed incubation diet, we see they're eating a lot of squid. So in Harrison's study, it was 50% fish eggs with some squid, and during incubation in my study, they're eating half their diet is squid. 22% of that is myctophids, and then 11% crustaceans and some others. Within the squid component, 40% are species that would suggest that they're using a post-spawn scavenge strategy. Whereas only 10% are species that they would be getting from more of a surface seizing, sit and wait strategy. So it's mostly looking like it's this strategy, but this is not, but it's important to say that this is not absent. They're still doing this to some degree. It's just a much more marginal strategy. Comparing the incubation to brooding diet, you can see that they're starting to incorporate different things into their diet. The squid component is reduced, and then now we get a huge influx of crustaceans and fish eggs, and the myctophids stay about the same in terms of importance. When you look at Laysan incubation diet, they're also just eating a lot of squid, and then supplementing that diet with some myctophids and crustaceans. And so if we look at this, so 60%, wait, let's go back to, okay. So I just want to talk a little bit about the implications of what this might mean. So if we're talking about mesopelagic prey, so it would be, okay, 50% minus 10%, so 40% plus myctophids, 62% plus crustaceans. So upward of 75% of their diet is coming from this mesopelagic prey base. And that's important because a lab mate of ours recently is doing work on mercury levels in elephant seals, and she's showing that, that the deeper the diver of an elephant seal, the more mercury is found in their system. So her work is indicating that there's a large amount of mercury in these deep waters, and these prey are actually serving as a vector of bringing mercury up into an ecosystem that you wouldn't expect it, like an albatross. Okay. When we jump from Laysan incubation diet to brooding, we're not seeing as much of a difference as we do when we see black-footed's diet shifting from incubation to brooding. So they're still eating a lot of squid. There's a little bit more crustaceans, but everything else is mostly the same. And again, another way to look at it, so just compared to each other, if we compare black-footed and Laysan incubation diet, it's not that much different. When we compare their brooding diet, though, there's a pretty big difference in the functional groups that they're targeting. So this is interesting to me because it says that maybe during incubation, when they're segregating by habitat, it's enough to sort of mitigate competition for resources. But when their habitats shrink and now there's a lot of overlap, Blackfoot seem to be adjusting, and now we see a lot more niche partitioning by diet. Okay, now to look at this a little bit more statistically, so this is an ordination. So I did a canonical analysis of principal components on this data, which basically it's an ordination technique that the output, you're basically looking for clusters to see how different these groups are in statistical space. So when I plot Laysan incubation and brooding diet, you see that there's not really differences between the two. There's a lot of overlap. They're not showing much clustering. They're not showing any clustering at all, which indicates that statistically the the diet is not different between incubation and brooding. But when I pop the Blackfoot data on there, the first thing you see is that there is a shift in this diet away from the Laysands, indicating that Blackfoot diet is different than Laysands. And also, the incubation diet seems to be clustering away from the brooding diet with some overlap, but there's definitely more clustering there. And to think about what that means in terms of what they're eating, you can overlay these species vectors. And I know there's a lot on the slide, so I'll just guide you through it. Basically, so look at this axis, and you have black-footed on this side, Laysand on this side, incubation down here, chick brood up there. And the way to interpret it is, so these prey arrows indicate which sort of trophic space they're in. So this area, these prey species influence black-footed incubation diet. So this particular mesopelagic squid, but in chick brood of blackfoots, it's mostly influenced, the differences are mostly influenced by these decapod shrimps, the pelagic red shrimps, and flying fish eggs. Whereas in Laysan, there's not much difference in, not really any spread between incubation and chick brood, which we saw with the different visualization. But the differences between the species are being driven by different species of squid, which again could go back to squid having very particular preferences of habitat based on sea surface temperatures. And so moving on from species differences, I was then interested in seeing how groups within species affected diet. So I looked at sex, and there were no differences between the sexes, so I just grouped everything. And then albatrosses are really long-lived birds. I'm sure most of you have heard about Wisdom, the albatross, who is 62 in breeding. So being such long-lived birds, age can be really important because these birds have a lifetime acquiring experience and memory that could influence every foraging decision that they make. So when I did the same thing for age, same kind of plot. Okay, so the younger birds are in bright green, and the older birds range from like middle-aged, light gray, to old birds, which are the black dots. And as you can see, there seems to be clustering of the young birds away from the older birds. And when I looked at what that meant in terms of diet, it appears to be that crustaceans and longlight-associated food are driving more of the differences in the younger birds' diet, and more of these North Pacific transition zone species like vertically migrating muscular squid from the transition zone, mesopelagic fish, so myctophids, and some of these smaller squid species are driving differences in the older bird age group. And then I did the same for blackfoots, and I didn't really see too much clustering. There's a big spread in the young birds and a big spread in the older birds. What's interesting, though, is that there's this little group right here that sort of pops out, but they seem to be an equal mix of ages. But when I look at the diet, the prey vectors on top of that, it's interesting that it appears that there is a group of birds, there's a group of blackfoots that appear to be specializing on myctophids, and it has nothing to do with their age or sex. What is also interesting, but not as strong of a trend as in the Laysands, is, so there is a lot of spread in the young birds, but they do seem to be more clustered over here. And when you look at the diet, again, crustaceans are also on that axis, and longline-associated bait, even though it's a weak, it's still associating towards that side of the axis. So potentially, younger birds are influenced by the same sort of, or the differences in the younger bird's diet is potentially influence the same across the species. So, I mean, I have no explanation for why, but potentially these are more marginal prey types, and so these younger birds are potentially getting excluded from more preferred prey and going after these more marginal foods. Okay, and then lastly, I wanted to look at dietary specialization. Again, albatrosses are really old aged birds, and so I thought that, like the sea otters, potentially individuals could have a preference for a particular prey and exhibit that preference over time. And just briefly to sort of understand how I measured that, I wanted to show this figure, and what it shows is basically this curve represents the diet of an entire population of animals. Underneath that curve, you see all these smaller curves, which represent, each curve represents the diet of an animal within that population. Now you see up here, there's a lot of overlap in all these individuals' diet curves, and they all look like the curve of the population. So that basically just means that individuals in this generalist population are all eating the same stuff. They're eating a lot of things, and everybody eats different things. But down here, you get a different kind of generalist population that is composed of individual specialists. So you still have the same niche width, composed of the same number of diet items, but in that population, you have individuals that aren't overlapping very much with each other, which means that as a whole, yes, they're eating a lot of different things, but individually, they're not eating very many things at all. They're more targeting certain prey types. And as you saw in the figures that I showed when I just showed the raw diet, yes, they're eating across all these functional groups, but as you can see, individuals are hitting up certain prey items really, really hard. And I know this is one sample taken from one bird. This is a sample taken from a bird once. But that diet sample represents diet incorporated over a long period of time, so weeks. So that represents multiple foraging events for that bird. So even though it's one sample, it's representing diet from over a long period of time, multiple foraging events. So if a bird is, showing a high percentage of myctophids, it's because it's been eating myctophids for 2 weeks straight and probably has a preference for that. So when back to here, you can measure this amount of specialization using this index. And so I did that, and what I found was, well, I was mostly interested in looking at how increased competition during chick brood affected this specialization index, because as we saw in sea otters, when competition increased, individual specialization also increased. And I actually am seeing a similar thing with the albatrosses, mostly in black-footeds, where it's more strong in black-footeds. But this axis, basically, on the left, you get a more specialized diet. On the right, it's a more generalized diet. And you see a shift from incubation to brooding, with brooding diet being more specialized. Same thing in the laysands, but there's a lot more overlap, so not as strong as seeing that shift in blackfoots. The other thing to note is that I should have made these axes the same, but the numbers are tiny, so I'll just have to talk you through it. But kind of the median value of blackfoots is around 0.5. If you look at the laysands, it's shifted to the right, so blackfoots would show up more here, indicating that blackfoots as a whole, as a population, have more individual specialists than do laysands. And potentially this could be, they have, during the incubation and chick brood period, especially during chick brood, they have more of a restricted range than do laysands. So both species do restrict their range, but blackfoots do it more than the laysands do. So potentially they're having to deal with greater interest-specific competition than our laysands. And then I wanted to look at the specialization index relative to age. And what I expected was that these older birds with more experience are probably going to have more of a preference for prey than younger birds, so they'll show a more specialized diet. But I found the exact opposite. And it's not really, it's mostly just in blackfoots again. But the younger birds are showing a more specialized diet than the adults across the breeding season. And so I've been trying to interpret what this might mean, and I don't know, but one reason might be that as a long-lived seabird, as an adult, you've acquired a lot of knowledge about the habitat you're in, about different places to go to forage. And so adults might be more prone to sort of cut their losses at a particular prey patch and be more willing to move on to the next prey patch because they know where other prey might be. Whereas a younger bird might be more reluctant to leave a prey patch because there's food there and they don't know if they're going to find food somewhere else. That's just complete conjecture. I have no idea, but it's just one potential reason for seeing this. For seeing that. So that's it. I actually don't have conclusion slides because I hate making conclusion slides, and I knew this talk was going to be kind of long. So I guess the last thing I just wanted to say is that this model is turning out to be an incredibly useful tool, not just for filling in some holes of traditional diet analyses, but also allowing us to answer some of these more complex questions about relationships between these birds and their environment and among themselves. I guess that's it. I'll leave you with this very awkward-looking image of an albatross. Any questions? Yeah.
Two questions. The first one has to do with the last part, where you talking about specialization, and especially during the different times of the seasons. Is there any indication from the tracking data how much overlap there is between the young and the older adult birds?
That's like I just ran this and now it's on my to-do. So I haven't looked at the age differences, but I definitely want to see if why I think maybe we're seeing that. I should see in tracking data that the adults have more movement, whereas the younger birds are more just having higher residence time in a certain patch. But I haven't looked at that yet, but I'm going to.
All right. So the second question has to do with your discussion in the first part about sea otters and how sea otters become specialists to some extent, and how females feed the pups a certain type of prey, and the pups then gain that experience eating that prey and become specialists on the prey that their mothers fed them over time. So, then you went on and talked about generalists and specialists later on. So I'm asking whether it'd be interesting, whether you actually think it's possible that the chicks take on a specialization based upon what the parents are feeding them.
For albatross, I think probably not. Mostly because the chicks are getting fed, like with the sea otter, they're holding it and they're seeing how the parents is tearing it apart. But for an albatross chick, it's being fed by its parent, it's just down the throat. So they don't even see what they're being fed. We do have some tracking data from fledglings, and it looks like they're foraging in completely different areas than the adults, so there doesn't seem to be any overlap with adults and young. Although, what's interesting with blackboots is that older birds have very distinct white butt patches, and I've always wondered if younger birds might cue into that and say, oh, there's an older bird, I'm going to follow that bird and see where it's going. But as far as we know, there's no sort of cultural transmission.
Yeah, I knew it. I expected it from taste, not so much.
Okay.
So
yeah, maybe. I mean, you hear it with humans. But yeah, I don't know.
Yeah, I was wondering about the bycatch interaction thing. It seems like you found that the lace sands were eating more like discards or offal of the boats, and maybe that the blackboots are eating the bait more. Yeah, and then also that the chicks might be targeting the bycatch more. So I was wondering if those trends have been seen like in the demographics of the bycatch.
Yeah, that's a really good question. So all of this stuff is fairly new, so there's been a lot of work, as you know, with longline fisheries bycatching, and so I think it'd be really useful to see if we're seeing the same kind of age differences. I'm not sure if they'd be able to tell what the birds are eating, whether it's the bait or the prey species, but they have observers, so they might have that data. And it'd be really nice to have more corroborating evidence so we can feel more confident that this model is working. Yeah.
I found it really interesting that you didn't find any difference difference between the sexes of albatross, considering, correct me if I'm wrong, but there are several studies showing the sexes utilize different areas of the Pacific, at least for lay stands, and I would expect that that might result in a dietary difference as well.
Yeah, I should really not say what I'm going to say, but I actually don't, I haven't read that the sexes have different distributions. So now everyone of you is lost in the years there. That's definitely, that is interesting. Maybe it's because this is all from the breeding season, so there are a lot more constraints on where they can go. During the rest of the year, they can go basically, well, not wherever they want, but there's a lot more freedoms where they can go, and you see it between colonies too. During non-breeding season, there's a lot of segregation between colonies. So I think non-breeding season there's probably more partitioning than during the breeding season when they're under a lot more pressure. But yeah, that's really the point, and I'm going to go with that.
Maybe one more question, and then we'll keep these things going.
So that's awesome. You have the data from the breeding season, but it makes me wonder, I'd like especially that question about what they're doing in non-breeding season. Would that be a viable project, or would the effort to catch them during non-breeding season be too much?
People do. People have caught them in the non-breeding season off boats, and the diet is, it's an interesting question because diet included, it's all about diet, like the timeline of the diet. So the stomach oil, I'm actually not sure if they would have stomach oil in the non-breeding season. It seems to be a strategy to help them keep them on the nest, because the alkalized oil usually get water, and so the thought is that they're really using this oil not just for sustenance, but when you're waiting for your mate for 3 to 4 weeks, you also need water. But even if they did, again, that's diet only over a couple weeks. So potentially you could sample their adipose tissue, and people have done this with their receiver tissues, and maybe get a longer integrated diet, which would be really interesting. But yeah, I don't know.